Friends, in this video I am going to discuss on recombinant DNA technology. As you know that this field is very emerging field. It came into existence in 1972 by the great work of S.N. Cohen and Herbert Bayer. Both these scientists were working on two different fields. Cohen was working that how plasmids, which is present in the bacteria, could provide resistance against an antibiotic, while Herbert Bayer was interested in knowing that what is the mechanism of action of restriction enzymes, how restriction enzymes work. When both these scientists met in a conference, they took the decision to solve their problem jointly, and finally when they worked together they came with this recombinant DNA technology. First of all, we have to understand what is the meaning of recombinant DNA technology. Recombinant, as you know, it means new combination. New combination means by taking two different things and bringing them in certain new combination. For example, you take that in English we have total 26 alphabets. By making different combinations, we can develop a large number of words. Similarly, in our body, in every organism, there are a large number of genes which are present in the form of DNA, that is deoxyribose nucleic acid. These genes can be presented in different combinations in different organisms naturally, and when the same combination is produced by artificial means, the technique by which these new combinations are produced is termed as recombinant DNA technology. In general, you call this as genetic recombination. It is the exchange of information between two DNA segments. Means when two different DNA segments come together and produce a new combination of genes. And you people also know that this is a very common occurrence within the same species. For example, when I will give you certain examples, you are well aware of Mendel's laws. You know that Mendel worked on bees, and he took two characters that of the bee, that is your yellow seeds and round seeds in one plant, and green seeds and wrinkled seeds in another plant. Okay, suppose this chromosome has genes responsible for green seed and wrinkled seed, and this part of the chromosome is responsible, or in you can say have the genes responsible for yellow seed and here your round seeds. Now when the meiotic division starts, then in the meiotic prophase first division, there is a crossing over between the homologous chromosomes, due to which in packaging certain parts of the chromosome break and rejoin in other homolog. In this way, some part of the gene is carried by one homolog, whereas its broken piece is carried by another homolog. In this way, the same two characters, that is yellow and round, green and wrinkled, now produces four different types of combinations, which you observe in the Mendel's experiment. Now we will come to the recombinant DNA technology. As far as definition is concerned, recombinant DNA technology can be defined as when a gene of one species is artificially transferred to another living organism, it is called recombinant DNA technology. Or it can also be defined as production of a unique DNA molecule by joining together two or more DNA fragments not normally associated with each other. Means जब हम दो different DNA segments को जो कि normally under natural conditions वो दोनों एक साथ नहीं आ सकते, उनको artificial तरीके से by manipulation we bring them together, the two different sequences of DNA. That technology is termed as recombinant DNA technology. It is also when this gene sequences which has been bring together are multiplied in great numbers, then that technology is termed as cloning, or you generally it is also termed as molecular cloning or gene cloning. For recombinant DNA technology, that is for the development of recombinant DNA molecule, we have to follow certain specific steps before finally getting the recombinant DNA molecule. Those steps are written here. That is identification and isolation of gene of interest. Means सबसे पहले तो आपको आइडेंटिफाई करना है कि what is the gene in which you are interested, and once you have identified the gene, you have to isolate that particular gene from the host chromosome or from the host genome. Once you have isolated the gene of interest, then you will form a gene construct. But there are many conditions when you could not isolate your gene of interest directly from the host genome. In that condition, you have to decode the protein which is responsible for that particular character in which you are interested. For example, suppose you are interested in the insulin gene and you are unable to find the exact location of the gene in the genome. In that condition, you have to decode the protein. Decode the protein means you have to identify the sequence of amino acids present in that protein, because you know that proteins are made up of amino acids. Once the sequence of amino acids has been deduced, then you know that every amino acid has assigned genetic code. On that basis, you can form the nucleotide chain which can form that sequence of amino acids. So once you have deduced the nucleotide sequence, from that sequence you can form the DNA molecule, and this DNA molecule which will be generated from protein is termed as cDNA. cDNA means complementary DNA. In further lectures we will explain what is complementary DNA. Now once we have found our gene of interest, either directly from the genome or by decoding the protein, we have to form a gene construct. Gene construct means that genetic component which has both a vector and the gene of interest. Later in the video we will come to know that what is gene construct. Once the gene construct has been developed, now there is a very important step, that is insertion of this gene construct into the host cell, in which you will multiply in numbers this gene, your gene of interest. Since you cannot observe this DNA molecule, that is gene construct, by your naked eyes, so you have to devise a method by which you can know that what are the cells in which your gene construct has been introduced, and that process is termed as identification of transformed host cells. Once you are assured that our cells has been transformed, then those cells will be selected and put in a chamber where for the amplification of the clone DNA, means for the multiplication of the DNA which you are going to clone. Now when you have multiplied those DNA sequences, then again you have to isolate the plasmids from bacteria and cleave it into linear cassettes. Means after isolation from the bacteria in which you have amplified the gene sequences, you have to separate the vector DNA and your gene of interest, and by cleaving and producing them a large number of those genes, and they are termed as linear cassettes. Now those linear cassettes can again be used for introduction in other organisms to produce transgenics, means new organisms with new characters. We will take all these steps one after another. First of all, we have to understand the whole process by which the recombinant DNA molecule is produced. This is the host genome from which we can find our target DNA, means our gene of interest. This is the gene of interest here selected, and this is the vector DNA, means the molecule which will be used for making the gene construct. Generally, the vector DNA is the plasmid molecule which is present in the prokaryotic organisms like bacteria. Now, this plasmid molecule is linearized by cleaving this plasmid molecule with the help of an restriction enzyme. The circular plasmid is now here, you can see that it has become a linear molecule. This is in fact the vector DNA, functions as vector DNA. Now the target DNA is separated from the host genome. Again, this is done by use of restriction enzymes. In this way, your gene of interest and vector DNA has been linearized. Now both these two molecules are bring together and joined in a single molecule. This is the part, blue portion is the vector DNA molecule, or you can say plasmid molecule, and the red portion is your gene of interest which has been selected from the host genome. Now when both these two molecules are joined together with the help of an enzyme called ligase, the molecule which develops is termed as DNA construct. Now this DNA construct is introduced in another host cell, here in bacterium, to multiply the number of these gene of interest molecules. Inside the bacterial plasmid, the conditions are provided so that these bacterial molecules can multiply. The insertion of gene construct into the host cell is termed as transformation. Now this is again a very crucial step by which we can introduce our gene of interest into the bacterial host cell, and it's at here you have to screen each host cell where this gene of interest with the vector molecule has introduced inside the host cell. Now we will take the same thing, but with a specific example, that is of growth hormone gene. How this growth hormone gene can be separated from a human. This is the human cell. Inside the human cell, a nucleus is present. The nucleus has the genetic material, the 46 chromosomes, which is a very vast molecule. When you see the length of this all these 46 chromosomes DNA. Now from that vast DNA, the growth hormone gene is separated, and this growth hormone gene is separated with the help of an enzyme called EcoR1. As I told you when in my another lecture, that is restriction enzyme, this EcoR1 has the ability to cut the DNA sequences and produce the sticky ends. Sticky ends means the two ends of the specific DNA sequence where it will produce a nick have overhangs, means have single ends, or you can simply say that is it becomes single stranded at that location where this EcoR1 enzyme cuts. And these DNA sequences are very specific nucleotide DNA sequences which is specific for a single restriction enzyme. Now the same restriction enzyme is used to cut the plasmid which is present in the bacterial cell. This is plasmid, this is the bacterial host chromosome, and this is plasmid which is circular. When this circular DNA molecule, that is plasmid, is cut by the EcoR1, then the same type of nick is produced, means single stranded DNA will be present at both the ends. Since both these single stranded DNA, which is termed as sticky ends, are overhangs, they are exact complementary to each other, so a DNA bond will be formed. When these two molecules are mixed together, then the DNA then hydrogen bonds are formed between these two segments of the DNA, that is growth hormone gene and bacterial plasmid. In this way, again a circular molecule is formed and the growth hormone gene get inserted. Now once this insertion of growth hormone gene in the factor molecule or plasmid molecule has taken place, this molecule is now termed as gene construct. This gene construct is now introduced again back into the bacterial cell. This process of introduction of gene construct into the bacterial cell is termed as transformation. This step is done so that we can multiply our gene of interest. Now, what are the enzymes which are involved in this recombinant DNA technology? The first enzyme which is required is lysozyme. Lysozyme enzyme is the enzyme which breaks the cell wall, means outermost layer of the bacterial cell. Ribonucleases, these are the enzymes which is used to digest the RNA, ribonucleic acid. Deoxyribonucleases, which is used to digest the extra DNA molecules. These deoxyribonucleases molecules are of many different types, like exonucleases, means those deoxyribonucleases which cut the double-stranded DNA from the two ends, either singly or in oligonucleotides. Endonucleases are those DNases which cut the DNA molecule from the middle, means they divide the DNA molecule into two parts. In these endonucleases, restriction enzymes are the most important. Of these restriction enzymes, the restriction enzyme type 2 is very important because they or you can say they act only a specific sequence where they can make or they can produce an egg, and finally the DNA ligase, which is used for joining the two different DNA molecules together. Many other enzymes also come into picture, but these are the main enzymes which have role in recombinant DNA technology. The restriction enzymes in the recombinant DNA technology are in fact termed as seizures. Seizures means catching, which is used to cut the DNA sequence at a specific you can say nucleotide sequence. For example, the EcoR1 requires GAATTC sequence. Then when it gets this gene sequence, then it cuts in between G and A towards the 5' end, and similarly here towards the 5' end it produces an egg at G and end, and these two ends get separated. As a result, in both these DNA molecules, certain part of the DNA molecule becomes unpaired, or you can say it becomes single stranded. Now DNA ligase enzyme also play a very important role. When the two different DNA molecules comes to get a chance to come closer to each other, the hydrogen bonds are formed between the complementary nucleotides, and once the hydrogen bonds are formed, the DNA ligase joins both these strands by formation of phosphodiester bond. So in this way, the two different DNA molecules which are present in two different organisms, two different individuals, can come closer to each other, and a new combination of molecule is formed. Recombinant DNA molecule, it requires a vector DNA. Vector DNA are those molecules in which the foreign gene can be inserted, and the main requirement for a DNA molecule to function as a vector, that it must have a origin point where from the replication is get initiated. It should have one antibiotic resistance DNA sequence, or you can say gene, by which you can easily detect the presence or absence of this particular plasmid molecule, and a specific gene sequence which can be detected by a restriction enzyme. For example, here you are seeing EcoR1 is detected when EcoR1 cleaves the DNA molecule only when a sequence GAATTC is present here. This is essential because only then you can insert your gene of interest, and this vector molecule can be used for cloning of your gene of interest. First, the different vectors have been used in rDNA technology. The first vector which was used was plasmid, which is present in the bacteria. Then these plasmids have certain limitations because they can carry very small DNA sequences. The lambda phase was used. Again, when certain larger DNA sequences was planned to clone, then the property of both plasmid and lambda was used, and that molecule is termed as cosmid. Then bacterial artificial chromosomes, yeast artificial chromosomes. In this way, there are many different types of vectors according to requirement of the scientist. is used as vectors. These bacterial artificial chromosomes and yeast artificial chromosomes can carry large genes can be inserted in these molecules. Expression vectors are those vectors in which when gene of interest is inserted, then the specific proteins which can be produced by the genes which has been inserted in those molecules. Similarly, there are subtle vectors. Subtle vectors are the vectors which can carry the gene of interest both in prokaryotes and eukaryotes. This is our requirement during the experiment. Now, this is the gene construct. Gene construct means here the gene of interest is present. Then if it has to act as a expression vector, then the promoter and report is also required. Then this is the sequence of plasmid, means the vector sequence, and this is the terminator gene which is responsible for termination of the transcription process, and these are the sites which are detected by the restriction enzyme. Now, how this gene construct can be inserted into the host cell? This is a very complicated process, and there are many mechanisms by which the gene construct can be inserted into the bacterial host cell, and one of those mechanisms is the electroporation. Electroporation means by the use of electricity we can create pores in the outer wall of the host cell, and once the pore is created, then our gene construct have a chance to get entry into the host cell. In this electroporation process, first of all, a very high voltage electricity is created for a very brief time, in milliseconds. Once this high electricity is created, then the pores develop into the outermost wall of the bacteria. As soon as these pores develop, the gene construct which is present in the surrounding medium have a chance to get entry inside the host cell. Some of the gene construct here you can see has entered inside, and as soon as this electricity is removed, again these pores automatically get blocked, you can say, and in this way the gene construct can be inserted inside the host molecule. Now, once the gene construct has been inserted into the host molecule, this process of insertion of the gene construct is termed as transformation. Here you can see that this is the bacterial host chromosome and this is the inserted gene construct. Now, though this process looks very easy, but in fact there are very few chances of getting a very successful insertion of gene construct, because there is a chance that the plasmid which has been cut open, here you can see the plasmid has been cut open by a restriction enzyme, instead of joining with the gene of interest, it again rejoins by itself and get introduced into the bacterial host cell. Then there is also chance that the gene of interest DNA segment get joined, its two ends get joined and form another plasmid like circular DNA structure and get introduced into the host cell. Now both these host cells which has been introduced with such type of DNA, means without inserted foreign DNA into the vector, is useless for recombinant DNA technology. Only those plasmids, those vectors which had the foreign DNA, the gene of interest, get inserted into the plasmid. When they get introduction inside the host cell, then only that host cell can be used for molecular cloning or recombinant DNA technology. To screen, to detect that what are the host cells in which the inserted vector is present, we have developed many techniques. Either here some resistant properties, antibiotic resistant properties of the bacteria is used, for example ampicillin, or a latest blue and white selection technology has been developed. White colonies, here you can see that two different types of bacterial colonies. Some are white and some are blue in color. The white colonies are with the inserts, means they have the gene construct with the foreign DNA, whereas the blue colonies have no inserts. This is by this color difference we can easily differentiate the host cells, or you can say bacteria, which have the inserted DNA molecule or which don't have the inserted DNA molecule. Now once we have isolated the DNA molecule which have the inserted gene of interest, now those bacteria are put in fermenters. In this type, this is the fermenter in which the favorable conditions can be artificially maintained for the growth of the bacteria. In these fermenters, or you can say bioreactors, we can easily multiply our bacterial culture in a very short duration. The product is continuously removed and purified. In this way, this is the whole tanker fermenter in which the bacterial culture is maintained. Once the bacteria has been cultured, now you can see that these are the plasma vector sequences in which our DNA inserts are present. So in this way, from a single DNA molecule, we have developed millions of copies of those inserts, or you can say foreign DNA, or you can say your gene of interest. And now once we have get the enough number of these molecules, this process is termed as gene cloning or molecular cloning, you can say. Once you have got the enough number of these molecules, now we have to separate those DNA molecules, and if that culture is used for production of the specific protein in which we are interested, then such vectors are termed as expression vectors. This is the you can say in short the recombinant DNA.